Hello, I hope you all like our new introduction. My name is Tom Bultrees, and I would like to welcome you to this Porous Media Tea Time Talk. I'm here today with Kamal Singh, Catherine Spuren, and Mohammed Nouraipur. And two other members of our organizing team are Maya Rucker and Marcel Moura, who unfortunately couldn't join us today. The Porous Media Tea Time Talks, to remind you, is meant to give a platform to junior porous media scientists, and is complementary to the geoscience and geoenergy talks in our featured channels. With that, I will hand over to Mohammed, who will chair our first talk of the day.
Thank you very much, Tom. Today's first presentation is by Filippo Miele. Filippo completed his master's in statistical physics at the University of Florence, focusing on pattern formation hosted on network type support. Then he did his PhD between the CIISc Institute in Barcelona and the University of Lausanne under the supervision of Professor Marco Dens and Professor Pietro Diana, where he performed experiments for studying the filtration of colloids and biocolloids. Today, he gave a presentation on the filtration by porous media. Thank you, Filippo, for joining us today, and the floor is yours.
Okay, so just to give a small introduction, filtration is basically a two-phase separation process where you want to separate the solid particle from the fluid phase where they are suspended. Now, among the three mechanisms that drive filtration, which are physical strainings, immobilization by stagnation zone, and absorption, we will focus on absorption as soon as it couples the two mechanisms of transport and reaction. Indeed, absorption occurs when particles are transported close to the surface of the grain or the solid matrix, and they interact between them. Now, the interaction is encoded in a general way by the DLVO theory, which tells you that once the particle gets really close to the surface of the grain, the overall energy profile that the particle felt, which is represented here from this solid line, comes from the balance between the attractive potential and the repulsive potential. Now, the particles can be filtrated, so permanent trapped, if it's able to cross the energy barrier. Now, the most successful attempt to describe filtration by coupling reaction and transport is represented by the Apple sphere model, which tells you that given a certain representative portion of your porous media, you can model it with a single sphere and a liquid envelope. Now, among all the particles that are flowing through the porous media, here represented by the area A2, only a small portion of them passing through the area of size A1 can get in contact with the collector, so with the filter. Now, the size of A1 is driven by gravity, Brownian motion, and the flow. Once the particle gets close to the surface, then the electrostatic interaction is triggered. The particle has non-zero probability to be attached. Now, in this framework, so the concentration of the retained, so attached particles named with A, is just proportional with the concentration of the still flowing particles. So the suspended particles here named with C. And the attachment rate, so the constant that gives you the retained particles, is nothing else than the ratio between the two areas, A1 and A2. Now, in this framework, so filtration by the porous media is just a sequence of scattering events. And the two main diagnostic microscopic quantities that describe filtration are the breakthrough curves, which is the distribution of arrival times at a given distance at the reference plane, and the deposition profile, which is the concentration of the attached particles along the distance of the filter. Now, the underlying hypothesis behind this theory is that after each collision, the concentration of the still survivors of the C suspended particles is homogenized. This means that the CFT, so the classical theory, assumes the existence of an REV, over which the particles are filtrated with a constant rate. Now, within this framework, so the classical framework, it's possible to describe filtration by the classical advection diffusion reaction equation, where you can see the terms of diffusion of the drift terms and the filtration rate. And it's quite easy to derive that in case that we can neglect dispersion, diffusion, which is the case for high Peccary number, so the deposition profile is supposed to decay as an exponential slope. Now, several other data collected in the lab that show that the deposition profile is not following an exponential decay. The hypothesis, which is the main hypothesis of my work, is that, okay, at this stage, we completely neglect the heterogeneity that comes from the flow, and so we want to investigate the role of such heterogeneity for the deposition profile. Now, what we start is that at the just level of the simulation, so we employ this 2D heterogeneity porous media, where the grain is represented by these gray circles, and this porous media is characterized by a given specific power law distribution for the pore size. Now, solving the Stokes equation, so we have the flow field here representing the color map, and then simulating a 2D Lagrangian trajectory, we saw that for this case, the velocity exhibits a strong spatial correlation, as you can see from detailing of the autocorrelation function of velocity. Now, if we define a criteria to measure such spatial correlation, is it possible to build the distribution of such spatial correlation? Now, within this framework, we can interpret filtration as a sequence of random events, where at each step, so the particles is crossing a given jump length of a distributed correlation length, is crossing this jump with a given distributed velocity, and it's subjected to a given attachment rate. Now, to validate our stochastic interpretation of filtration, we need an experimental setup that allows us to make both microscopic observation of breakthrough curve and deposition profile, at the same time also the microscopic pore scale analysis. So that's why we decide to go for a microfluidics approach. So here you can see the scheme of the chip that we employ for our experiment. So the total length is a bit less than 1 meter. And again, the pore size has been characterized by the Delaunay triangulation, and it follows a power law distribution for the pore size. Here on the bottom, you have an idea of the size of the chip which fits in one end, and then we perform experiment by fluorescence microscopy of both deposition profile and breakthrough curves. So the flow system was represented in this way. So we collect by time-lapse image acquisition at the outlet the concentration of the still flowing particles in order to measure the time dependency of the breakthrough curves. And then for the deposition profile, we scan in 28, but it can be extended to whatever is the number of the point that you want to sample. We sample, so in 28 different positions, the deposition profile. Here you see an example of the flow system. So we continuously inject particles, so fluorescent particles. And then the flow rate that we choose was in order to have a mean velocity of 150 around microns per second, which correspond to a Peccary number of 10 to the power 4. So we use a magnification 6x for the objective, and the colloidal suspension, we in order to avoid gravity effect at this stage, so we use a density matched suspension by adding heavy water to just pure water. And in this case, for this preliminary experiment, we work at 0 ionic strength. So this means that we didn't add any salt to the colloidal suspension. So just to have a look directly at the data, we saw that if you look at the breakthrough curves, working at 0 ionic strength, 91% of the particle has been recovered at the outlet of the filter. And it's more interesting in looking at the complementary breakthrough curves, which is defined like 1 minus the current concentration divided by the injected concentration, which show a tailing, a power law tailing behavior with the arrival times with a characteristic exponent of minus 0.63. While with the same experimental setup, we measured the deposition profile. And here you can see that after 30 hours of injection, even working at 0 ionic strength, we were able to collect a deposition profile. So we are in front of a clear pattern deposition profile, and it seems to be an anomalous deposition profile. As soon as it has this power law behavior for more than 2 orders of magnitude, and then it exhibits these strange peaks close to the outlet. Now, the advantage of our experimental setup is that it's possible to find the link between the macroscopic observation of the breakthrough curves and the deposition profile with the post-case process by particle tracking. Now, the complexity of this confined, heterogeneous confined geometry requires that we needed to write from zero our particle tracking scheme. For which reason? Here you can see that while the deposition profile and the breakthrough curves has been collected in fluorescence channels, so we were able to detect only particles, for the particle tracking, as soon as the trajectory they are compressed and stretched, we have to work in phase contrast microscopy. So this means that this allowed us that we collect at a higher frame rate, so 60 frame rate per second, with 2 millisecond exposure times. And here you can see the grains on an average size of 60 microns, and the colloids that are represented with these black dots that are flowing. Now, as I said before, the complexity of such systems require to write from zero the particle tracking. Why? I will briefly show the reason of such challenging tracking. So, usually, most of the algorithms that are employed in tracking particles are based by the fact that for each frame, you assign coordinates to the particle that you are able to detect. Then you have to link the candidates, so you have to link the pair coordinate between 2 consequent frames. So the most so in this case, you can see that your brain will intuitively assign the following particles at a given frame, and then the next one and the next one. Now, to translate what your brain is doing in just an algorithm, most of the algorithms, they are using the nearest neighbor criteria, which means that at each step, at a given frame, you are searching for the closest candidates in the next frame, and in that case will be the best candidate. Now, you can see that if this works, for example, for this case, the criteria may fail in case that you have 2 or more candidates, so 2 or more closer particles within a given distance. And if you are using the criteria of minimizing the distance, you may also assign the wrong candidate to such particles. Now, to overcome this problem, we decide to by the fact that we acquire image at a quite high fast rate acquisition, and that makes the trajectory of the particles was pretty smooth, we decide to change the algorithm of the particle tracking in this way. So given by the first pay, we were able to somehow compute the local velocity that the particle feels. Then we make a prediction of where the particle could be in the next frame, and then we are searching for a candidate with a new predicted location in a distance which is smaller than the one that the nearest neighbor required. Now, with this particle tracking scheme, then we were able to collect and track particles over 3 different fields of view, which means that we collect particles, we track particles for 6 minutes in one field of view, then we move the state, and we collect particles for other 6 minutes, and then going on until and then you go on until you reach the maximum distance in trajectory that you want to observe. Here you can see the result of the 3 sticked field of view, and here you see the details and the quality of our particle tracking, and at this stage you can see the effect of the diffusion. Now, by reconstructing the Lagrangian trajectory, we define a criteria to measure the correlation length. And so the correlation length is the pieces along a single trajectory over which the particle get closer at a given distance to the closest grain. So for our experimental setup, we decided this threshold distance was fixed with 2 microns. So by identifying the point where the particle get close enough to the grains, we put a flag. Then between 2 flags, for us, this is a correlation length. Now, sampling this correlation length of an arbitrary number of particles that has been tracked, and then by deriving the transition velocity of that the particle needed to cross this correlation length, we were able to beat the PDF of correlation length, velocity df, and attachment rate. Now, for the PDF, so we see that the PDF that we measure by our particle tracking gave us rotation PDF, so correlation length with a negative slope of the exponent minus 0.4. So I want to stress a bit between the meaning of this power law distributed correlation length, which somehow breaks the concept of the existence of a single REV, so a single length scale. Now, for the by the same particle tracking, we were able to measure the velocity PDF, and then for the attachment rate, we employ a result that has been derived and measured by science and collaborators, where they measured for a straight channel a deposition profile of that scale with an exponent of minus 0.5 and attachment rate that decays with so it corresponds with an attachment rate that decays with this characteristic exponent. Now that we built our PDF, we are able to run our stochastic model. So the stochastic model is written in this way. So at each step, you draw a given jump length, correlation length, and a given velocity. By the ratio between the jump length and the velocity given, driven by the PDF, we can compute a transient time, so another vector time. Then, by a given value of the attachment rate, we can compute a filtration times. So now the competition between the advective time, that is the time that the particle needed to cross to make that jump, and the filtration times, the times that the particle has to make this jump, tells you the fate of the particles. Now, in case that the advective time is below the decay time, so the particles survive, and then you update each time the new position. While if the transient time is higher than the filtration times, the particle is considered deposited, and the deposition profile is computed. If the particles reach the outlet, then the BTC in that case, so the Bretton curve, is then computed. Now, just having a look at the comparison between the CTRW, so the simulation that we did with experimental data, we saw that for the PDF that we use in the first case, the simulation are able to reproduce the early transient behavior of the Bretton curves, but they completely fails in describing the long-tail behavior of the Bretton curves. And this is typical of quite, let's say, homogeneous porous media, which means that you are allowed to average on the velocity and just consider the mean velocity. And this is quite obvious as soon as if we look at the PDF that we were able to measure with the particle tracking. Of course, it shows a peak on the mean velocity, while the low velocity, the extremely low velocity, they are quite unlikely to be chosen. Then, on the other side, we saw that if we look at the deposition profile, so the comparison between our model and the deposition profile, we observe that the model is not able to catch the large distance deposition profile, which comes from the fact that the correlation length that we sample, of course, the maximum distance was the maximum distance that we were able to collect was given by the number of the field of view that where we performed the particle tracking. So we were in front of two problems. So the first is that so our particle tracking is affected by finite time windows, because of course we cannot track particles for an infinite amount of time. And then, so in our case, we were tracking particles for just 6 minutes. And then the other problem is that the particle tracking is affected by finite space windows, which means that the maximum, theoretically, the maximum correlation length that you can measure is the number of field of view that along you track the particles. So to solve the problems, in a theoretical paper that that I previously published, that show how the deposition profile is affected by the three random variables, we saw that in this case, to overcome the undersampling of low velocity, we derived that the early distance deposition profile, so these slopes, is mainly driven by the slope. So actually, it has exactly the same slope of the power law velocity PDF. While on the other case, to overcome the problem of the finite size space of the space window, the correlation length, we extend the correlation length PDF with the maximum length of our chip. Now, by changing the slope of the low velocity for the velocity PDF and just extending the range of the correlation length, so the relation PDF, by keeping the same slope, we got a much better agreement between our simulation and the data. So indeed, the model tends to a bit overpredict the early arrival times, but it completely describe the tailing behavior of the breakthrough hoods, and the agreement, and we get a better agreement for the deposition profile, while the strange peak close to the outlets is still, let's say, under investigation. And with this, I conclude the presentation, and I thank you for the attention.
Thank you, Filippo, for this wonderful talk. I would like to ask our audience if they have any questions and comments. Please post on your right-hand side window. We got first question from Catherine. She's asking, how challenging would it be to convert your approach to 3D? If you can, I think, visualize all the stuff through the.
Okay, so actually, I just applied for a fellowship to to go to Davis and then try to make the same analysis on a real 3D porous material. So the thing is that in that case, we reproduce a real soil rock sample, so there is a real structures. Then we will make exactly the same, we will follow the same scheme. We will solve the flow, we will simulate the Lagrangian trajectory, so we will measure again the correlation length, and with the column, we will compare with the deposition profile and breakthrough hoods. So actually, I mean, this is exactly the next step that we are going to do, that I'm going to do if I will get the fellowship.
Sure. Good luck with that. I've got a quick question. It's probably naive or let's say more technical. You showed the experimental case, which is actually impressive and complex at the same time. The design is there. So you used a Packley number of 4. I think there was a certain flow rate there. I was wondering if, because you're tracking particles there, is there any higher velocity like limit you can track the particle with certain accuracy?
Yeah, clearly. So indeed, this is the mean velocity, but then of course, due to the distribution of pore size, you have so the higher velocity is could be 10 or 50 or 100 times the mean velocity. So there is a limitation of the particle tracking, and then in that case you have to find a good compromise between the mean velocity, which is something that you can tune with the syringe pump, so by imposing the flow rate, and also the let's say the concentration of the colloidal suspension. So if you want to go faster, then it's better to use a low concentration. So in that case, you are increasing the interparticle distance, and then you can tune the parameter where to find the best candidate between two frames. So so basically this is a parameter of the particle tracking that it tells you which is the which is the maximum distance over which you may find two candidates, which is given by the maximum velocity and the frame rate at which you are collecting particles.
Okay, wonderful. Thank you very much. Anybody has question from the studio? If not, I would like to thank you, Filippo, for this wonderful talk, and thank you for coming to the studio and joining us today.
Thank you.
And we wish you good luck for your future career and also for your grant, for the fellowship grant.
Okay, thank you.
Okay, so let's move on to our second speaker, Saurabh Singh. He's from Indian Institute of Science, Bangalore, and he's recently defended his PhD thesis. He's worked on granular materials and particularly cemented granular materials. So he currently works at IISc, Indian Institute of Science, and works on mechanics of soils. So today he's gonna present his work on termiteness, which is quite close to me as well, because we've been doing this work only for about 2 years, and this is a very fascinating topic. So I'm looking forward to it, and thank you again, Saurabh, for joining us today. And the floor is yours.
Thanks, Kamal. So, good evening. My name is Saurabh, and I'm going to present our work on termite mounds. The title of my presentation is Living Porous Media, Biology and Engineering of Termite Mound Construction. This is the outline of my presentation. So first I'll introduce animal beta structure, in that termite mounds and termites, and then I'll enlist the objective of this study, following which I'll talk about the results which I've got from our experiments, and then I'll conclude the presentation. Now, coming to the animal beta structure. So in nature, we found animal beta structure around us all the times, and these structures are used for different purposes, such as for shelter, for protection from predators, for catching prey, for reproduction, and for thermal regulation and ventilation. The material used in these structures could be exogenous material, or it could be endogenous material, or it could be combination of these. Exogenous material is the material which is coming from outside of the species, that is collected material such as mud, wood, leaves, and feathers. Endogenous material is the self-secretion from the insect or from the animal, and the hybrid material is, as I told you, is the combination of the two. In case of termite mounds, the mounds are used by termite for shelter and thermoregulation and ventilation purposes. Now, these termites are found across the globe near the tropical and subtropical regions, such as in Australia, India, Africa, South America. Now, coming to the termites are highly social insects which have evolved from cockroaches. There are more than 3,000 known species of termites around the world, and a fraction of them make termite mounds, and a subset of these will further grow, cultivate a fungus farming garden inside the termite mound, which will be used for food. Now, there are three castes of termite mound. There's a major worker, minor worker, and soldier. The major and minor worker do most of the work, and soldier is basically protecting the colony. The termite mound can be thought as extended phenotype of termites. The size of the mound is 3 orders of magnitude larger than the termite. So a termite would be of size, let's say, a few mm, and the termite mound would be a few meters. The termite mound can endure several decades, much longer than the life of the termite or termite colony. It provides a controlled environment for termites, and it is also useful for thermal regulation for fungus garden. So here there's this fungal comb that is at the base of the termite mound, and this is the food that they use to eat. Okay, now coming to the process of construction of termite mound. So basically, termite takes the nearby soil and they form small boluses. These boluses are basic building block of the termite mound. They go and deposit it on the site. And because of the consolidation process and wetting drying cycle, and so basically the mound is constructed, and the structure of the mound is controlled by gravity, wetting drying, and remolding in the case when there is a breach by an external agent. So now coming to the objective of this study, so it is known that the termite mounds have high strength and also good ventilation. These two objectives are sort of competing in one sense. So we here try to understand how they are achieving this competing objective. So we study the termite mound at different cross sections. So we take the different cross sections of the mounds at different heights and study the variation of their mechanical properties or their hydraulic properties. So we further study the stability of the mound slope. So in this study, we have used two mounds. One was abandoned mound, that is dead mound. There was no termites in it. And the other was live mound. In case of abandoned mound, we have sectioned it at different cross sections at different heights. And our focus is primarily to understand the behavior or the properties distribution along the cross section. So we have identified two things. One is the buttress, which is outer wall of the termite mound, and then the core, which is inner region inside the mound. So we have also studied on the live mound. In the case of live mound, the samples were extracted. This was not sectioned, and the samples were extracted at two heights, 0.9 meter and 1.2 meter from the base. Now, so we perform very simple experiments. So first of all, we do a uniaxial compression test on a micro universal testing machine. So because the specimen size is restricted to around 10 to 20 mm, so we obtain a cylindrical specimen of size 10 mm cross 20 mm height. And for porosity, we use X-ray computed tomography. And then for air permeability, we have a custom-made glass T-tube. So I'll just tell you about the custom-made glass T-tube. So in this one, we have a porous, we have a pressure transducer. And at one end of the glass T-tube, we have sealed our mound sample. And from the other end, we are supplying the air. So basically, permeability is measured through an indicator. The indicator is the pressure required to maintain a certain velocity. Let's say I have maintained certain velocity, what is the amount of pressure required? So through that, we are measuring the permeability of air through this media. Okay, now coming to the results. So, the test was performed at core and buttress at different heights. So if you see in the abandoned mound, you can clearly see that the core has higher strength in comparison to the buttress at around all the heights, for both in case of abandoned mound and in case of live mound also, we see quite a bit of difference between core and the buttress. So it tells us that the mound is a bilayered structure. It has a strong core and a weaker buttress. Now coming to the porosity, so basically we have taken samples and put it in CT, and so to get the porosity distribution, we have binarized this X-ray CT volume, and then in this binary volume, we have two phases, whites and soil, and we are throwing random cubes at different locations, and we are measuring the porosity of each cube, and through that we have obtained the porosity distribution. So if you look at the porosity distribution at different heights, you could clearly make out that, so the red plot is for core and blue plot is for the buttress. So at the top of the mound, you have higher porosity and the buttress, and so top and middle of the mound buttress is more porous, and then the core at the bottom of the mound core has higher pore sizes in comparison to buttress. So this again tells us that the core at the top and middle region is much more denser than the buttress. Now coming to the air permeability, so here I'm showing you pressure versus flow velocity plot. So as you can see that for at different heights, so A2, A4, A7 are different heights. For each height, we will require more pressure to maintain certain velocity for the core in comparison to buttress. As you can see here, like blue plot versus green plot. And for all the heights, we have immaterial of height, or at every place we have seen that the core is much more is relatively impermeable in comparison to the buttress. So this tells us that the termite mound has a stronger core and a porous buttress, which the permeability of the buttress is also higher. Now, we have also performed the slope stability analysis. So this termite mound can be idealized into two shapes. It could be a triangular or it could be a trapezoid. So we have taken it as an axis symmetric case here, and so as we have observed that the buttress and core have different properties in our analysis, slope stability analysis, we vary the properties as obtained from our experiments along the cross section. So we linearly interpolate the properties, and so for calculating the stability of slope, I use a strength reduction factor method. So I'm using Moore-Coulomb criteria for identification of failure. And each time I'm reducing the strength by a certain factor. And when there's a contiguous failure surface formed throughout this geometry, we identify that as a failure, and the corresponding factor by which I am dividing the strength is our factor of safety. Now, if I consider the base infinitely rigid and take the properties I have obtained from the experiments for the upper region, I found that the factor of safety of 46 for trapezoidal mound and factor of safety of 113 for the triangular mound. This tells us that the reality should lie in between somewhere 46 to 113, but in reality the base of the mound is not infinitely rigid. So actually, when I consider the properties of the base of mound similar to whatever neighboring soil has, so I found that if a mound of 15-meter height could be constructed if the factor of safety of 1 is considered. So that is quite tall mound. We don't see it in nature, but this is the capability of the mound strength that we can have. So in the man-made structures, we usually, for ordinary structure, the factor of safety is considered between 1.5 to 2, and for dams, let's say it's 3. For a nuclear power plant, it could go for up to 10, but nowhere in human construction we have a factor of safety of 46 and 113. So that's like a really high stability. It could be because the termite mound has to be safe from the other animals, reptiles, and so on. So they might require much more stable or safe shelter. Okay, so now I'll conclude my presentation. So we have found that the mound is a bilayered structure, and through this bilayering, which basically you have a porous buttress and much denser core, it is able to achieve a temperature and relative. It works as a stabilizer for temperature and relative humidity. So there was one of the studies in our mechanical engineering department where they've shown that if you have layered structure in which you have a porous, first layer is porous and the bottom layer is much denser, the evaporation sort of stops, and it controls the whole environment. So through the bilayered structure, termite mound are able to achieve a good ventilation, and because of a denser core, it is able to achieve a high strength. So thank you.
Thank you, Saurabh, for this wonderful presentation. I would like to mention that everyone who has a question can type it in the comment section. I have a question of my own. But when it comes to factor of safety for the slopes, how you measure soil shear strengths and stresses for different samples of mound, and do you think when you analyze different mounds in different parts of world, can you see a normal distribution for this factor of safety or a
So
range
the
for them?
factor
Or
of safety is a function of the material properties here. So these were like basically. So we are using a more Coulomb failure envelope in this. So basically we have cohesion and a friction part for the soil. We have obtained it from our uniaxial compression testing, and there were some direct shear tests also that were performed. So from there we get this factor of safety. So the factor of safety is high because the strength is much higher than the size of the mound. So for this much size, we do not require the termite soil to have this much that high strength. So I cannot actually say about the distribution of factor of safety for all the mounts. But if I have the properties of all the mounts and the distribution, I can do this slope stability analysis and get it. But from the two mounts that we have tested, we found that the results are quite consistent, and I don't think there's any issue. In saying that the factor of safety is quite high for most of the termite mounts.
Thank you. We have a question from Raihana Akbari. Do you have any measurements to characterize mineralogy, grain size of tested specimens for strengths, any chemical analysis of tested mount samples, any contribution
so we
from
have
such
grain size
parameters?
distribution. So this study was done by like a few researchers. So one of my senior did the initial analysis, and this would be in 2016 by Khanda Sami. So you could see search for termite slope stability analysis on termite mound. So there you could find that he has done the grain size distribution. For the chemical part, we have done the IISc. So it's basically there are two things that come in the mound soil. The one is basically the surrounding soil, and the other is termite secretion. We don't see any other chemicals in the mound soil. Yeah. I hope this answers the question.
Okay. Perfect. And the last question from Josh Labaki. The crazy high safety factor is very puzzling. I would expect natural structures like that to have evolved to be more material saving than ours, not less.
So basically, most natural structures are optimum in some sense. So if you see trees there, they go trapezoidal, the base is much broader than the top part. But termite mound, here the stability analysis is from the human perspective. I have only considered the loads of the self weight. But that's what I was telling, that there could be a breach from a reptile or something from an external agent. So mound has to be safe from that. It's not just self-fit. So I think they are optimum in their design. I don't think they are putting extra effort. But it's not just so this slope stability analysis is as we do for human structures, and that's why it is high. We cannot. So if we indent on it, we'll see that indentation is also, it will require some force. It won't be so easy.
Okay. And the last question. There seems to be a difference in material properties between the live and dead mounts. Any ideas about that?
It's not, the difference is not so great. Let me go back to the presentation. So if you see the abandoned mound is left, there's nothing going on. There's the termites are not living in there. in case of live mount, what is happening is that and time upon time again time, they basically, if there was any breach, if there was rain, and so they rebuild the whole thing. So, so there's any breach in the mount, they will rebuild it and the properties will change. If there was a dead mine which is left from, let's say, 5 years, so the properties might not. So basically, it's getting cured and all those things are happening. There's nothing going on in the inside the mount. So that could be one factor which is affecting the strength. And I don't see there's a great difference in the strength parameters though.
Okay. Any question from the studio?
Yeah, I had a short question. So if you look at the micro CT scan, it seems like there's quite a bit of sub-resolution porosity. So probably multi-scale structure. I think Kamal's work also showed this. Do you have any comments on that? What I mean, does that have any functionality in the
Definitely there will be. Yeah, so we could not from this. So our CT scan had a resolution of 13 micron. So the pores lesser than that size would not be identified here. We had access for mercury-induced porosity, but the thing was that it did not. It just stopped functioning, and we could not get that analysis done. Otherwise, we could have got pores till in nanometer sizes. So in that sense, this analysis is restricted to only macro pores. It's not detailed. Yeah. So in that sense, it's not fully. That's I hope that was your question. If otherwise, you could repeat it. I don't know. Yeah.
No, that's fine. Thanks.
Mohammed, can I ask? Not ask, it's a comment in fact. So when we did our studies on these were different mounts. We were fascinated with the fact that we found porosity, connected porosity, let's say about 25%, and we had permeability of 1 Darcy, which is 10⁻12 meter square. But the fascinating thing about your study here, which is a different mound, I think fungus growing mound, your porosity is quite high.
Yeah.
It's like 40 to 50%. It's really crazy high, and I think permeability is high. I'm very curious to see what you can do with the micro CT image later. It's not a question, I'm just putting as a comment. If you can run simulations on it, again, resolving it a slightly higher resolution, like Tom said, with the including multi-scale factor from nanoscale imaging to the larger scale imaging and putting all the flow.
Yeah, so
I'm very impressed.
I think if we had the access to nanopores, we could get the connected porosity and then tortuosity and everything. That would have been much better. But yeah, this is what we have. Right?
Yeah. No, this is fantastic. And thank you for the presentation.
Okay, wonderful. I think now we can wrap up the session. Thank you once again, guys, for your time and these great talks. We wish you good luck with your research and your career. Our next session in 2 weeks will be given by, will be given by Mamta Jotkar on chemically driven convective dissolution. And the second presentation will be given by Ying-Da Wang on deep learning and efficient simulation on digital rock analysis. I would like to thank you, our audience, again for your support, comments, and suggestions. Please subscribe to our channel. Please contact us at porosmediattt@gmail.com and subscribe to our mailing list. With this, we will say goodbye, and we hope to see you in 2 weeks. Bye-bye.